The overall goal of this procedure is to acquire high-quality total internal reflection fluorescence, or TIRF images of cell wall-bearing microorganisms. This is accomplished by first preparing cover slips and cells. The second step is to precisely align the microscope setup for optimal image quality. Next, the correct incidence angles and imaging conditions are chosen for acquisition of images or movies. The final step is post-imaging processing of the acquired images. Ultimately, TIRF microscopy is used to study the dynamics of protein localization at the cell cortex.
The main advantage of this technique over existing methods, like conventional or confocal fluorescence microscopy, is that the image contrast in TIRF microscopy is very high, allowing fast acquisition times and low light dosage.
This method can help answer key questions in the field of membrane research, such as the mechanisms of lateral protein segregation or the mobility of membrane proteins.
Cover slips for TIRF microscopy should be cleaned from dust particles prior to use. Since TIRF is sensitive to background signals arising from dust on a dirty cover slip, a cleaned cover slip with less background is required. To begin the procedure for cleaning cover slips, use forceps to place cover slips in a ceramic holder with a lid. Next, fill a glass container with 1 molar sodium hydroxide. This sodium hydroxide can be reused multiple times. Place the ceramic holder containing the cover slips into